So let's talk about reproduction means of prokaryotes. Prokaryotes can reproduce asexually. Asexual reproduction comes in the form of binary fission. Basically, this is going to be when bacteria clones itself. And the process of binary fission is very similar to mitosis. So we're talking about a single-celled organism here. So we're wanting to go from one cell to two cells. You're trying to make a clone of yourself. That's mitosis, in a sense. It is going to be different because we don't have a nucleus, and so the way that that DNA gets allocated and the way that that DNA goes to its opposite pole is going to be a little bit different compared to how much mitosis occurs in the eukaryotic cell. But the end product, what I want you to see here is that the end product are two genetically identical cells. This is asexual reproduction in prokaryotes. Binary fission can occur quite quickly. What was one bacteria becomes many bacteria, and this is why they're able to be so successful by means of adaptation in different environments. They reproduce quite quickly. Budding is another asexual reproductive means of prokaryotes. In budding, we have a small formation seen on the parent cell. This is a bud, and that bud eventually grows. Nucleus copies over, genetic material gets transferred to that bud area, and that bud eventually comes off of the parent. It doesn't really leave the parent, so this isn't binary fission, and we have a complete new organism forming after that bud has finished growing, but we actually have a separate daughter cell, and then that daughter cell then buds. A new bud can form in different areas, and more budding events occur to where we start seeing multiplication of that organism. This is called budding. It produces chains of cells, and we can actually see how the cells like to stick together in the process of budding. We have sexual means of reproduction concerning prokaryotes. We actually have 3 different kinds of sexual reproductive events in prokaryotes. The first type is transformation. Transformation occurs when a prokaryote uptakes naked DNA. So here I can see a donor cell, and I have some DNA. There's in a nucleoid region, that blue area. There's all the genetic material. I've highlighted this red portion here. That red portion then gets released along with some other bits of DNA and actually gets uptaken by another bacteria that's located nearby. Bacteria like to be close together. And so that DNA is uptaken by another bacteria and gets incorporated into their nucleoid region. That process is called transformation, the uptake of naked DNA. Transduction occurs when that gene of interest, I still have my same donor, gets infected by a virus. Viruses that infect prokaryotes. Prokaryotes can have viruses, just like humans can. Prokaryotes can have viruses. The virus is called a phage. And the phage comes, takes up the DNA by random, not necessarily because it wants that DNA, and then it infects another recipient cell. That recipient cell will get that DNA. This is called transduction. DNA is transferred by the virus. The virus that infects bacteria is known as a phage. Conjugation is when DNA is transferred between bacterial cells in the form of a plasmid. This is the true mating of 2 different prokaryotes, because we actually see a fusion occurring between the 2 different organisms, the 2 cells, and the plasmid, seen by the circular structure here, gets transferred. Prokaryotes, I didn't discuss this in detail, but prokaryotes have plasmids. Plasmids is another area that has other genes of interest in a prokaryote. Well, that can get transferred to another prokaryote. That is called conjugation. It's a true form of mating. All 3 types are sexual reproductive events in bacteria. In prokaryotes. So just to reiterate, we have 2 different types of gene transfer here. We have vertical gene transfer and horizontal gene transfer. In vertical gene transfer, so vertical going from up to down, we're going from parent to daughter. And I can actually see that the parent produces genetically identical daughter cells. This is called vertical gene transfer. So this is binary fission This is budding. These are asexual reproductive events. Then in prokaryotes, you can have horizontal gene transfer, so going from right to left in this image. This is transformation, transduction, conjugation, where we actually have the genetic material not being completely identical because we have gene transfer going on in these three different events. These are sexual reproductive events in prokaryotes.